bunch of people were like itching to like actually understand it, so I reordered his slides because Dr. Thomas really likes Christopher Nolan and wants to be him. But he wants to lecture as Christopher Nolan. So I reordered his slides, and then in the middle of the lecture, like the not lecture, the review, there's like a couple slides that are out of order, but those are the ones where again maybe just drawn up. Just memorize the facts about those, you'll be fine for the test. So, and then I'm going to do up until the end of week 3, which is gastrulation, and then Aziz is going to do week 4 and the stomite stuff. Cool? All right. So, the first thing we're going to talk about is this stuff at the end. So this should have been the first slide of the lecture, but it wasn't. Right. So he draws this up, and then first fact you need to know that's actually not on this slide, but you need to know it, is that fertilization happens in the ampulla of the oviduct. Okay? Ampulla is that little part that like bends around, kind of opens up, right? And so fertilization happens in the ampulla of the oviduct. I remember that being a question on my test 2 years ago, like where does fertilization happen? I don't know why they care so much about that, but whatever. Fertilization, ampulla oviduct, it happens. After fertilization happens, a bunch of stuff happens that we don't care about yet, but Dr. Chilton will come in and like rock your world with it because she goes in detail about it. You get the zygote, and then you start having these little stages depending on the days you're on. So for the first 2 days, right, you have a zygote first day, and then day 2, you divide into 2 cells, and at this point, the thing that it's dividing into is called a blastomere. I doubt they're going to ask you that, but just in case, mention on there. Blastomere divides again, goes to a 4-cell stage, and you're still on day 2, and then you get to this 8-cell stage, and then here is where you start going compaction. So they might ask you when does compaction happen, and it happens day 2. Cool? After compaction happens, you go to a morula, which he said in lecture that is 16 cells. So once you get 16 cells is when you get a morula. Everybody cool with that? Alright. So far pretty simple, nothing too complicated yet. So he jumps from that to this, which is just 1 day anyways, but that's fine. So essentially what he has drawn here is a blastocyst, and he has the outside part is called the outer cell mass, or trophoblast, same thing. And then the inner cell mass, or the embryoblast. Okay? So the outside part, the green, and then the blue part that's right here is the embryoblast. Okay? Inner cell mass. Okay. So really important to note on here also is that he has written down here is that the zona pellucida has broken down. So zona pellucida like degenerates in this stage. And then another important thing to note is that the blastocele forming is actually what makes it go from a morula to a blastocyst. So it goes from collection of all the cells, it's right here day 3, to a blastocyst because you get this little cavity in the middle of it. Does that make sense? Okay, so far simple stuff. He jumps to day 8. I don't know why, but day 8. So the book, I checked just in case because I was like, there's no way it's day 8. The book says that implantation begins at the end of week 1, but he specifically verbally said in lecture that beginning of implantation is day 8. So just around the end of week 1, day 8-ish is when implantation begins, and that is the end of week 1. Week 1 is very easy, very boring. The facts I told you about that like he highlights in the lecture are like the important ones to know. Everything else is too detailed. Don't learn anything else about it. Cool? The second week is called the week of 2s. The reason why is because you get a bilaminar germ disc, which he has written up here, and then you also get your trophoblast splitting into 2 different things. So what it means by week of 2s is your inner cell mass will become 2 different layers, and then your trophoblast will also become 2 different layers. That's what week of 2 means. Okay? So this is kind of like misleading in the way it's drawn because he has this like thin part drawn right here, and you would lead you to believe that this is actually what's implanting, but it's not. This top part where my arrow is pointing, which is where it would actually be implanting up here. Okay? So it implants on the side of the ICM. Okay, so the inner cell mass is where the actual blastocyst will implant into the uterine wall. So he doesn't really like straight up explain any of this stuff, so it doesn't matter like how it happens. All you need to know about this part is that you have your outer cell mass turns into what's called a cytotrophoblast, so that's one of the layers that your trophoblast turns into. And then the other one being the syncytiotrophoblast, right? We haven't seen it yet, but that's the other layer that it turns into. And then your epiblast, right here, this blue part, and your hypoblast come from what was your inner cell mass or your embryoblast. Does that make sense? So your embryoblast, inner cell mass, turns into this guy. Epiblast, hypoblast. Okay? Your epiblast, this blue part, is what becomes the embryo. It doesn't only become the embryo, but that's where all of the embryo comes from. Your hypoblast does a lot of other stuff, but it doesn't become the embryo. Okay? And then you still have your blastocyst right here, this big opening from what took you from a morula to a blastocyst. Okay? Alright. So day 9, the blastocyst is invaginating even more into the uterine wall. And your syncytiotrophoblast, this layer that's out here, he doesn't have it highlighted on here, but I drew an arrow. So the next couple of slides are all going to be the exact same picture. It's just that he highlights different parts and labels them, which to me is confusing because he doesn't like talk about them right away. But hopefully if I talk about them first and then go over them slowly, you'll understand them. But the cytotrophoblast is one-celled white layer right here. Do y'all see that? Can y'all see the arrow on here? Okay, there you go. So the white layer that's of one cells, those are the part of the trophoblast, right? That's one of them. And then the other part, the syncytiotrophoblast, is right here. The outside part looks like a network of stuff. And what's essentially happening is these single-celled cytotrophoblasts are like mashing together, and they're making more of themselves, and they're becoming the syncytiotrophoblast. So syncytium means like a network. And you have multinucleated cells right here that are burrowing their way into the uterine wall. So what actually burrows its way into the uterine wall, what like has the enzymes to do it, is the syncytiotrophoblast. And I did not label this specifically, I just drew an arrow on purpose, or a line I guess, but I wanted you guys to like try to reason this out. Does anyone know what this is? Leukinae. Okay, anyone know what leukinae are? Pools of blood, from what specifically? But like what blood vessels specifically? From the uterine arteries, okay? So like uterine blood vessels, okay? So what happens is syncytiotrophoblast buries into the uterine wall, and doing so causes the blood vessels that are in the uterine wall to break down and form pools of blood. Okay? That's all it is. Lacunae. Lacunae. Okay? So the week of 2's issue with like 2 different layers, those are your 2 layers from the trophoblast. The 2 you get are syncytiotrophoblast, which is out here, and then cytotrophoblast, which is right here. Another thing to note, in case they ask you about it, I don't know if they will, but if they ask, HCG, human chorionic gonadotropin, is released from the cytotrophoblastic layer. I don't know if they'll ask you that, but just in case. HCG is associated with cytotrophoblast. Okay? So inside, where you have your inner cell mass, where you had your epiblast and hypoblast. The epiblast doesn't really do anything yet. It's kind of just still sitting there. And inside this little like cavity that forms inside of it is called the amniotic cavity. He makes a big deal to stress this, and I don't know if it's because he's going to ask a question about it or why, but knowing that helps differentiate the difference between the twins very easily. And I'll show you when we get to the twins part why it makes so much sense. But the amniotic cavity forms inside of the epiblast. So it forms essentially inside of the embryo. Okay? So remember that. Amniotic cavity inside embryo. And then the hypoblast, this bottom layer of cells that's extraembryonic, right, will start to migrate down this way, right? So before it started migrating, what was this area called? It's called the blastocyst, right? It was right there, right? So now this yellow layer of cells is migrating downward to connect and close off what was originally called the blastocyst. It'll no longer be called the blastocyst. Does that make sense to everyone? Okay. So they're coming down this way, and they're coming down, and they're going to connect. Okay? So we're still at day 9 right now. Day 8 was this one? Day 8 is this one. 9, sorry. Okay? And when it closes all the way, what's inside of there is called the primary yolk sac or primitive yolk sac, either one you want to think of. Okay? because it's going to go away. And then the secondary one comes along and does this thing. But anyone have any questions yet, or are we good? Okay.
Hey, Adam, so do we need more specific days, like this is day 9, or is it more just like the second week?
It's more important to know that it's in the second week, because if they ask you questions about the days, it won't be, is this day 8 or 9? It'll be, is this day 9 or 14? Or like, is this day 9, not 14, day 9 or like 17, or like day 9 or like 24, you know what I mean? So you have to know, you know that if you count the days, that 9 would fall in the second week. Does that make sense? Okay. That's more important. Would you agree with that, Ben?
Yeah.
Yeah, okay. Alright. So this is the exact same picture that I had before, but instead of having the epiblast and hypoblast highlighted, he just has the amniotic cavity and primary yolk sac highlighted. So, that makes sense, everybody? Okay. Same exact picture again, just instead of having other things highlighted, he has the syncytial trophoblast and cytotrophoblast happen. Cool? So week of 2s, he went from, you know, 1 layer of everything and one blastocele to 2 layers of trophoblast, cyto and syncytio, trophoblast, 2 layers inside of the ICM, which are the epiblast and hypoblast, and 2 cavities, which is amniotic and primary yolk sac. Everyone cool with that? Okay. So we're still going along. Day 9. Day 12, what starts to happen is these cells right here, the red ones, are called your extra embryonic mesoderm. Okay? These will start to push out from the epiblast. So extra embryonic mesoderm come from the epiblast. Okay? And they will start to migrate into the space between where you had your cells from your, here, I'll just go back to this original picture. So you see these yellow cells that are coming down? So this space right now, there's going to be a potential space, right there, okay? And these cells are going to potentially push this yellow cell inward to make space. And it's going to do so. One is going to go against these cytotrophoblasts, like so, and the other one's going to go against the actual hypoblastic cells themselves. The hypoblastic cells that came out are considered extra embryonic endoderm. So don't get that confused. Extra embryonic mesoderm comes from the epiblast, and extra embryonic endoderm comes from the hypoblast. Everyone cool with that? Okay. Here's day 13. So it went from day 12, it started doing it. Day 13, the extra embryonic mesoderm has entirely surrounded the entire inner cell mass, which was the inner cell mass. So it surrounded all of the epiblast and the hypoblast, okay? All entirely surrounded by now, okay? That's why it was doing what it was doing with 1 layer going. So y'all can imagine if this went all the way around right here and this continued, it would end up looking like this, right? So what you have now is 3 different cavities. So you have this one up here, which we already had, which was the amniotic cavity. We have this one, which is the primary yolk sac. Then you have this guy right here, which is the chorion. Okay? Chorionic cavity. So if you look, there's all 3 of them labeled. Amniotic cavity, primary yolk sac, chorionic cavity. Everybody cool with that? Okay. Go along more. So then what happens after that is these extra embryonic endodermal cells will start to push out of the actual cavity that they formed. So these cells right here that form this cavity that my arrow's in will start to push into the cavity that was just formed. And new hypoblastic cells will start to push out from the hypoblast again and make a new cavity the exact same way, and then the old one pushes in here and then buds off, and then essentially just goes away. The primary yolk sac just buds off like this, and you see like a zoomed out picture right here. It buds off and goes away, and then the new thing that was formed is called the secondary yolk sac. Everyone with me so far? Okay. Okay. Yeah, we can go. So the cells that were making up the primary yolk sac, so these cells right here, okay, which were the extra embryonic endodermal cells, right? These will start to push out into the chorionic cavity. They will start to essentially just like stretch out into there. Okay, and while doing that, imagine pushing like one balloon into another balloon essentially. Okay? I know it's like hard to imagine, but if you had like one that was a little bit flatter than the other, and you push the full balloon into it, this flat one will start to envelop it. Does that make sense? Yeah? That's okay. Sorry. And then, so while that's going on, the hypoblast will send out a different layer, this yellow layer right here, which are new cells, to make another cavity essentially. And the old one, which was all right here, which was the old cavity, buds off, and then eventually buds off entirely, and then will join up with the, what's the cytotrophoblast, and then it'll just go away. So we don't have to worry about it anymore. So the primary yolk sac goes away.
So in that first picture on the left, are those two cavities the split chorion cavity?
This is the primary yolk sac pushing into the chorion. So see these on the left and the right? This was going all the way around here, but this pushed into it. So essentially split it in half, but once this goes away, it'll again be one big cavity. Does that make sense? No. It just goes. It just like small. It's a small. It just disappears. It just disappears. The chorionic cavity is still lined by extra endodermal mesoderm. Extra embryonic mesoderm. Okay. So look. So we had the extra embryonic mesoderm come around here, make this last cavity. When it made it, this right here was called the chorionic cavity, right? So what's it lined by? It's lined by extra embryonic mesoderm. Does that make sense? The area where the primary yolk sac goes to is like inconsequential, it's like small.
So you're saying the big cavity is the chorionic cavity?
This one is not chorionic cavity, yeah. So this is not chorionic cavity. This is secondary yolk sac, it's labeled. And then this right here is still your amniotic cavity. Okay? You're confused? I can go over it, it's like 100 times, I don't care. No, it's okay, I can go over it again. Tell me what you're confused and I'll go over it. I'll come find you after. Okay, good.
Wait, so the chorionic space is never actually mixed with anything else?
No, the chorionic cavity is not made of anything. The most inside layer of the chorionic cavity, okay, is made from extra embryonic mesoderm. Okay? The small little yolk sac that pinched off and embedded inside of it just goes away entirely. It just disappears. Like forget it even existed. Okay? Huh?
Sorry.
You're good?
Those two on the top, left and right,
This right here.
Yeah.
Sorry. What's on the left right here and what's on the right? You see my arrows? Okay. This is what was the chorionic cavity. This right here is pushing into the chorionic cavity, so it's essentially just occupying the middle space of the chorionic cavity. If you were to imagine this closing off right here, and then this circle coming in and just invaginating into this wall, you would have another space here, right? Because these would connect. Does that make sense or no?
So
They're temporary. It's temporarily just like occupied by something, and then it just balls off and then it goes away. Okay? I promise you these are very minute details that won't be important for the test yet.
So when it pushes down, what are those new cells called? Is that important or not?
Just hypoplastic cells. Hypoplastic cells, yeah. Okay. Any other questions? Good. I know it's a little confusing, but I promise you it's not that. This is still worth like day 14-ish. Okay? So like day 13 was when the extra embryonic mesoderm entirely developed it. But this is like day 13 and then you got day 14. And it's gonna say 14, 15, and what it means by that is like the end of the second week. Okay? So now we have 3 cavities. I don't know if we have the drop on this. He does, okay. So this is, now we have 3 cavities. So this is the remnants of the primary yolk sac. Does that make sense, everybody? Went away. And then you have your amniotic cavity and your chorionic cavity. Okay? Anyone confused? Okay. And one thing also that I don't think I mentioned yet. I haven't mentioned it yet. Okay, good. I haven't mentioned it yet? Yeah.
So the chorionic cavity is extra embryonic?
Yes.
Okay.
So because it was made by what? Extra embryonic mesoderm. Okay? Does that make sense? Okay. But it don't want you guys to think that the chorion is only made of extraembryonic mesoderm. It's just, that's what made the cavity itself. I'll tell you guys what actually makes the chorion in just a second, okay? But the chorionic cavity itself, this thing right here, was made because these cells pushed out from the epiblast and formed the way they did, okay? So that's what you're left with. This arrow's kind of in the way, but y'all see this little connection that's like, this thing's kind of like dangling from? This little connection right here, okay? That is made from extraembryonic mesoderm, and I think it comes up in a later slide, but I just want to make you aware of what it is. And that is called the connecting stalk, and that is what eventually turns into your umbilical cord, okay?
What's it made of?
Extraembryonic mesoderm. I think he has a picture of it. He does actually. It's labeled. There you go. Connecting stalk. So it tells you what everything is labeled. And he did everyone a huge favor here and like tell you chorion. This layer, extraembryonic mesoderm, cytotrophoblast, and syncytiotrophoblast, all three layers together are considered the chorion. Does that make sense? The cavity itself was formed because the extraembryonic mesoderm pushed out and formed around this guy and formed all the way around here. Okay? But the actual chorion itself is all three layers. Okay? So it's extraembryonic, does that make sense? Because the embryo is right here. This top circle you see, those are where our hypoplastic cells are, and this bottom circle is our hypoplastic cells. So what's this cavity that my arrow is in right now? Secondary yolk sac. And this one is amniotic cavity. Anyone confused? If you're confused, you can tell me. I'll go over it again. The chorion is all three of these layers: extraembryonic mesoderm, cytotrophoblast, and syncytiotrophoblast. And then it has labeled lacunae, but you guys know what lacunae are now. Unless you haven't already know it. The active thing is with different cavities labeled. Cool. Okay, so here's where he just like randomly jumped. This is where the lecture started, which makes no sense, but okay, so I just randomly stuck this in here just to explain a fact to you guys. You don't need to actually be able to visually understand this, okay? Just memorize some facts and you'll be fine for the test, I promise you, okay? But in the attempt to explain it to you, the chorionic, hopefully I can explain it to you, I don't know, hopefully I can. But anyway, amniotic cavity is this thing surrounding the baby that's developing, right? And the chorionic thing is the one that's surrounding that. Does that make sense to everybody? Okay. So then this collection of cells right here is what was our syncytiotrophoblast, cytotrophoblast, and extraembryonic mesoderm, okay? This right here, if you can imagine all this stuff going on, as it's getting bigger, it's taking up the actual uterine cavity. Does that make sense? So that's why you have this uterine space or uterine cavity. Does that make sense to everybody? As it gets bigger and bigger, it gets so big that it eventually takes up the entire space. And once that happens, two cell layers will fuse and close it off entirely. Okay? So, here's a picture of it getting bigger and bigger. Even though he had this one first, he had this one second. Here's it getting bigger and bigger, the space is getting smaller, and y'all see now the chorionic cavity is closed. Y'all see that? The chorionic cavity is gone. And this layer right here, called decidua capsularis, and this layer right here, decidua parietalis, when these two fuse, you no longer have your uterine cavity. So right now they're not fused, you still have a little bit of cavity. It doesn't matter what day this is because he doesn't tell anybody. He just started off with, here's an embryo, or here's a baby, basically. And he didn't really say much about decidua basalis, it's not really that important. So once those two fuse, they'll start fusing like this, and then eventually you'll have this structure. So you'll have the amnion, which is the thing surrounding the amniotic cavity, which is what the baby's floating in. You'll have the chorion, but you no longer have a space between it and the amnion, right? Because it fused. From here to here, it fused, right? Everyone sees that? It closed off? Okay? Question? Okay. And there you go. Cool? And then here is, he says the baby, what it looks like before it's born, but that doesn't really matter. But, does everyone understand that then? So the things you need to remember about this, like the highest of yields, I guess, is that the decidua capsularis and the decidua parietalis fusing closes your uterine cavity. Okay? Alright. So, after getting those facts down, I'm just gonna go back and make sure that everyone understands. What I just went through before the pictures of the babies popped up is the end of the second week. So essentially forming the secondary yolk sac is like the last major thing we know that happens in the second week, okay? So week 2 is over, okay? So the week of 2s is over. We're moving on to now what's called gastrulation, or the week of 3s. So now you have 3 things that are gonna form. I guess before I do that, I'll go over twins then. Okay, so we talked about in class the difference between all 3 or 4, I guess, types of twins, and he had them on different slides, so I just combined them, and I'm gonna walk you guys through it, so I don't keep looking through slides, and that way you can compare them. I think it's easier this way, so dizygotic twins are where you have 2 different eggs that are fertilized, okay? So you'll have 2 trophoblasts, 1, 2, 2 inner cell masses, 1, 2, which will form 2 entirely separate babies. Does that make sense to everybody? That's the easiest one usually for everybody. Okay. Monozygotic twins split at the 2-cell stage. So at the second day after fertilization happens, monozygotic twins split. Has anything happened in terms of differentiating the cells yet at that time? No. So essentially what happens is you end up having the exact same thing as if you had 2 different eggs. Okay? So essentially this starts off with 2 different eggs. This one starts off with 1 that basically splits into 2 different eggs. Okay? Everyone get that? Those 2 are the easiest. So monozygotic twins, type 1. I don't know if he actually makes you memorize it. Did you make you memorize the actual types? Like what they are? I don't know. I guess memorize it just in case. But monozygotic twins, stage 1, or type 1, splits at the 2-cell stage. Okay? And that's what happens. Monozygotic twins, type 2, is where you split like during the blastocyst formation. Okay? So when the blastocyst forms, the trophoblast, the outside layer, the outer cell mass, OCM, forms first, and then the ICM forms afterwards. So if the outer cell mass forms, and then the inner cell mass hasn't formed yet, and there is a split, you will end up having 1 trophoblast, but 2 inner cell masses. And since we said that the embryo itself comes from the inner cell masses, right, because that's where your epiblast comes from, you will have 2 amnions, but your chorion was made from over here, so you only have 1, and you'll have 1 placenta. Does that make sense? It makes it easier if you know like where all the stuff comes from, I guess. I don't know why you didn't. I don't know, I can't stop harping this, but anyway. So monozygotic twins 2, you have 2 inner cell masses, 2 amnions, 1 chorion, 1 placenta. Okay? This one, you have 2 inner cell masses, but you also have 2 trophoblasts. So this one only had 1, this has 2 for both of these. So you end up with 2 of everything. Okay? So 2 across the board, 2 inner cell masses, 1 trophoblast leads to 2 amnions, 1 chorion, 1 placenta. Okay? So you can pretty much always associate the number of inner cell masses you have with the number of amnions you have. Okay? Alright. So the last one, monozygotic twins 3, which is the thing we had on our quiz. And see, that's the kind of question he'll ask you. He won't just say, it's split after the formation of ICM. He'll ask you a question like, it's split at day 14, and he expects you to know that by day 14, the inner cell mass has already formed, right? Because the inner cell mass formed around day, 4-ish, remember? Before the whole implantation thing even happened. So, if that happens, the inner cell mass itself won't split entirely, it'll half split. But since you started off with 1 of everything, then you're starting to develop into 1 of everything, but then your inner cell mass splits kind of in half, and so you'll end up with conjoined twins. Right? So you still have 1 inner cell mass, but it kind of splits into 2 little separate ones, even though they're still conjoined. So you end up with conjoined twins.
Of these 4 options, did you ever say which is most common?
I think either dizygotic or monozygotic 1 is the most common. I'm not sure. Does anyone know? I don't know. I'm not sure. I don't think he asked you. We won't ask you that. Okay. That's your twins? Okay. So, everyone with me so far? Anyone confused or anything about anything so far at all? Any questions? Okay, no questions. Okay. So, towards the end of the second week, so that he has day 13 here. I don't know if it's important to know the exact detail of day 13, but towards the end of the second week. So this is showing you the same thing. This picture and this picture are the exact same picture. Okay? I don't have them that close to each other, so you can't see them that quickly. But this one, extra embryonic mesoderm, that's labeled right there. And this is the exact same picture. He just doesn't have the chorionic cavity labeled here. It's all this out here. Does that make sense to everyone? So this right here would be originally primary yolk sac, but then it becomes secondary. And this right here is the amniotic cavity. Okay? So this right here, the outside layer, is extra embryonic mesoderm. Everyone hold that? And this right here is the connecting stalk. Epiblast cells, and then these are hypoblast cells. Okay? So he orients you for you to understand like which direction, so that when you rotate it, it makes sense. And anterior, cranial, cephalic, rostral, all the same thing. Dorsal, ventral are opposing, and then posterior, caudal are different. The reason why dorsal, ventral, and posterior and anterior are like used interchangeably in humans is because we walk upright. These are developed in things that don't walk upright, and so anterior is towards their head, and ventral is towards their stomach. So ventral is towards the ground. Okay, so imagine like a lizard. Does that make sense? That's why it's confusing. Question? Good? All right. So what's going to happen right here is we're looking at a sagittal section of this. Okay, everyone from anatomy should know what a sagittal section is. It's essentially where you take something and cut it parallel to the midline. Okay, so if you were to cut my body like this, and then look into my body this way, sagittal section. Okay, so this would be the same as you guys looking into my body if I was cut right here and you were looking at me. Okay, so what he does in this next picture is he takes what's essentially the top of my head and he rotates it towards you. Does that make sense? Okay. It looks like that. Does that confuse anybody? Okay. So this was anterior. Dorsal is now facing out of the screen, and then ventral would be deep into the screen. Okay? And then posterior caudal is back.
I'm sorry.
You're okay. No problem, no problem. Okay. So, I'm trying to think of how to explain this better. Okay, so imagine a lizard. Okay? And I'm laying on my stomach right now. Okay? This part that says anterior cranial cephalic would be the top of my head, and then what says dorsal would be my back. Okay? The next slide, all he's doing is taking what is my back and then rotating towards you. So it'd be like I rotated onto my right side. Does that make sense? Because if you look at think about that. So essentially this ball of cells is like rotating out of the screen towards you, so that you're looking at it from top down. So all you can see, you see this like light blue layer right here, this blue layer? You're going to be inside the amniotic cavity looking at the top of that blue layer, which is that. Does that make sense?
Yeah.
Okay. So this is right, this is left, anterior, cranial, cephalic, rostral. You guys get the gist on that? Okay. So we're starting week 3. Okay? So this thing right here forms. I don't think it has, is that a label? Yeah, it has a label on it. Okay. So this thing right here starts to form, and this thing right here starts to form. This thing is called the oropharyngeal membrane. It's right here. And then the primitive streak and the primitive node grow caudally to anteriorly like this. Primitive node is just like an opening, and I'll explain what they are in a second. But they grow in the epiplastic cells, right? Because we're in the epiplastic cells. So if you imagine 3D, and you imagine like a space that's like right outside the screen a little bit, that's where the amniotic cavity would be. Does that make sense to everybody? Okay. I like to think, I know it's an extra word to remember, but it makes things easier for me. The oropharyngeal membrane right here is also called the prechordal plate, and it makes things a lot easier coming up in a little bit, and I'll explain to you why it makes things easier. But if you remember that prechordal plate, oropharyngeal membrane, makes things easier, okay? Oropharyngeal membrane, follicular membrane, primitive streak, primitive node. Okay. Essentially what happens is cells that are in the epiblast start to just essentially die out, and this just stretches out this way and forms like a gap in between these cells. Okay, that's what happens. The colloidal membrane is where the epiblastic cells and the hypoblast meet and fuse. The oropharyngeal membrane is where the epiblast and the hypoblast meet and fuse. So imagine two pieces of paper touching each other, and then you took like two staples and stapled one on one side and one on the other side, and you have like a gap in between them, so you can separate them out in the middle, but on the right and left side they're stuck together. Does that make sense? That's what happens. And this extra slide is unnecessary. So it looks like kind of like this. Does that make sense, everybody? So it's stapled right here and here, but in the middle you can take the papers apart. Okay, I'll get to that slide in a second, but okay. So you have this now, and this is, he says, the beginning of morphogenesis. So the generation of the structure of the embryo and formation of all three germ layers is gastrulation. Okay, so week of 3, third week. He talks about 3 germ layers. Someone today told me something really easy to remember, ectoderm by. They said that ectoderm is all the things that you find attractive in people. So if you're into sweat glands, like the skin and the mammary glands, I get. The eyes, ears, and nose, I get that. But it's actually pretty helpful. And that was Kaylee, for everybody who wants to know. Kaylee. Right. Okay. Endoderm is pretty easy. Endoderm is essentially your viscera, your organs. Okay. And then lining of certain things, like lining of your nervous system. And then mesoderm is like your muscle, your bones. I like to think of connective tissue just in general, and it's really easy to remember that way because blood is a connective tissue. We were talking about earlier, it's like a really good way to like pick up girls at parties. Like, you know your blood's a connective tissue? I don't need. Alright. Alright. So you can kind of get a general idea of what these things form. And here's a picture of how all this stuff starts happening. So the cells that are right here, okay, will start moving towards this gap that was formed in them, okay? And in doing so, they will start to spread out under where the endoplastic cells are, and they will start occupying this space that is formed in between the hypoblast and the epiblast. Everyone understand that? Okay. The first cells that do so, the ingressing cells, will go in here, and they will just essentially go as deep as possible into where the hypoblast is, and they will invade the hypoblast. You have a picture of it? He does, okay. And I'll go back to how this happens in a second, but they'll invade the hypoblast like this, and they'll embed inside of them, and then they'll just replace them, and that becomes your endoderm. So you no longer have hypoblastic cells right here once this all happens, okay? The next couple of cells that do it are the ones that become mesoderm. They will occupy the space, they'll go left and right, but they will go all the way deep as possible. And they will just fill in all the space in between where the ectoderm and endoderm are. Okay? All of this is happening because these cells that make up your epiblast and hypoblast are called epithelial cells. They're like lining of something, and they're very structured, but they have some signal that comes onto them that you don't have to know for the test. I don't even know what it is. And they're called mesenchymal cells. When they get this signal, they lose their structure and they become free-floating cells that can do whatever they want, essentially. And this is called EMT. I don't remember him asking a question, but it seems like something testable. Like, what is EMT? And he said something about in adults, this is like a common way that cancers work. So the epithelial cells will lose their structure and they'll migrate, do their thing. Okay? This is happening. Okay, so here's a sagittal view of this. So here, let me go back to where this was. Okay. So the first thing he did to make this easier for you guys is he took something and he cut this in cross section, like right where my mouse is going to go right now. So he cut it like this, okay? And then he made you look into it this way. So from where my arrow is now, he like looking in this way. Does that make sense? So if you look right here, you're looking into it. Okay? He then turned it to the side, and now you're looking at it sagittally. Okay? Does that confuse anybody? I know it kind of confuses me spatially so I can understand. Okay. So now you have mesoderm all in the middle, ectoderm on top, and then you have your endodermal cells here, which were your hypoblast. Okay? But they didn't come from the hypoblast, they came from your epiblast. They just migrated down and invaded and turned into endoderm. Here's where your oropharyngeal membrane is, where your epiblast and your hypoblast are touching. Here's your cloacal one. This one becomes your mouth. Oropharyngeal, you can reason that out. This one becomes your anus, cloacal. And so that's all happened so far, and we're still in the 3rd week, right? So this is all day 16 kind of stuff, okay? The last thing that happens that we care about for this part is the formation of the notochord. This is very high yield, I don't know why I like this so much, but the notochord becomes eventually the nucleus pulposus. Okay, I think he talks about it. Does he talk about it in section lecture? Okay, I'll just bring it up now just in case, because it's such a high yield thing. Nucleus pulposus is the remnant of the notochord. Okay? So notochord does not become your spinal cord. Please don't make that mistake. So you still have this streak open up top, and where the actual primitive node was, the biggest part of it in the front part, you have all of these cells ingressing in here, so all these cells are doing this. Where the biggest part is, right here, you'll have special type cells that also invade in there. They're part of the epiblast. You don't have to worry about what's special about them. They're just part of the epiblast, and they'll invade the biggest part, and they will only invade in this direction. So this right here is cranial or anterior. This is dorsal. This is ventral, and this is caudal or posterior. Okay, so you're looking at a sagittal view still, and this finger-like projection right here will invade in here, and this is called the notochordal process. Okay? From the primitive node. What do you mean comes from it? Yeah, so this hole right here that you're looking at is where the primitive node would be. You're right. Yeah, okay, thanks for remembering that. So this is where the notochord would be. Okay? This right here behind it, so posterior to it would be this regular mesoderm. This in front of it is called precordial mesoderm. And I told you guys earlier that if you remember that the oropharyngeal membrane is called the precordial plate, it makes it easy to remember that because this is called precordial mesoderm. So it's next to the precordial plate, but it's also precordial because it's before the notochord. So it's like, it makes sense on 2 different fronts. And it's precordial because it was made before the notochord, so it's like a 3rd reason why it's pre. Okay? So really hammered in there. Alright, so this notochordal process starts going in there like a projection, notochordal process, sorry. And it keeps elongating, so now you're looking at it from the top. So here's it from the side, here's it from the top, okay? And it keeps elongating, and then it'll eventually bud off entirely from where the primitive node is, okay? And then it'll eventually form this tube after it buds off, called the notochordal process. I think it was called, yeah, it's called at the beginning of it anyway. So it's a notochordal process, so it's a hollow tube now, and it's just kind of floating around in there. This does not become your notochord. I don't know why this happens, it just seems unnecessary. But it fuses with your endoderm. Yeah, it fuses with the endoderm, and it makes what's called a notochordal plate. And then after doing that, it just leaves again and becomes your notochord proper. But it's still made of the same type cells. And I haven't told you guys that yet because I don't want anyone confused. The cells it's made of are mesodermal cells. Okay? So mesodermal cells make your notochord. You can see they're mesodermal because they're labeled red. It helps you. Okay? Anyone confused yet? Or at all?
When the notochordal process happened, those are mesodermal cells. Is it because they underwent epithelial to mesenchymal transition, or?
The mechanism is supposed to be the same way. It's just the cells themselves are signaled somehow, which is about beyond the scope of the class as to like why they did that. I have no idea why. I just know they're like special mesodermal cells that make the notochord itself.
Okay.
But it makes sense that mesoderm is the one that makes it because the mesoderm is what makes your body and your connective tissue and your muscles, and nucleus pulposus is part of like your vertebral structure, so it kind of makes sense to think of it that way. Okay, so now we have the notochord, and that's it for gastrulation. So week 3 is now over. Next thing coming up is when Ben talks about it. So I'm going to talk about some clinical correlates that he mentioned a little bit, and then I'll have a couple questions questions that I made, see if you guys like understood what I was talking about, hopefully. And then Ben will take over and do his part, okay? Talking about teratomas, these are tumors that are derived from all three germ layers before they specialize into the individual parts that they are. So they're essentially tumors that come from the epiblast, okay? And they are tumors that can have teeth and hair, and I don't know if you guys have ever seen them before, or seen pictures of them, but they're pretty interesting looking, they're gross, but they're cool looking. And he specifically talked about a certain type, which are these sacrococcygeal teratomas. And he said it's pretty common for such a weird thing to have. Like 1 in 35,000 is not that common, but that's still, I don't know, something to worry about, I guess. And he said epiblastic cells that from the primitive streak in the sacrococcygeal region that did not die. So I told you guys earlier that when the primitive streak formed, those cells died, right? That's how it's made. So these are ones that did not die, and instead proliferated, okay? And that's why you end up getting this sacrococcygeal tumor. So those cells that are supposed to just drop in and then die so you can have the actual streak, they didn't actually die, okay? The next thing he talks about is this thing called sirenomelia. Siren, like the sirens from like Odysseus. They were mermaids, and it's easy to remember that because you have funky looking legs if you have this problem. And I apologize for anyone that has a family member or anyone they know that has this for me saying that, but it's pretty weird looking. And it's caused by reduced mesoderm in the caudal region of the embryo. So you have reduced mesoderm right here, which also makes sense if you think about it, because precordial mesoderm is towards your head. This is your notochord, so this is essentially going to be where your vertebral column is, correct? And everything under that, mesoderm-wise, is what forms your legs in terms of bones and muscles. And this didn't form properly because you don't have enough, so you have weird-looking legs. They're like fused. Okay? You can like make sense of it, I guess. And that's it. Okay? And I have a couple questions. Anyone have any questions before I show you my questions? Yes?
Alright, so is sirenomelia a type of teratoma, or is it something else?
No, it's something entirely different. The type of teratoma he gave you in class was sacrococcygeal teratomas. Okay? And the name of them kind of tells you where they are: sacrococcygeal, sacral area, coccygeal area. Okay?
Not quite important, but like, if it's like a blob in their face for moving, or?
I have no idea, honestly. I have no idea. All right. So I don't have any of the actual answer choices on here highlighted in terms of getting it right, because I want to talk to you guys through them. Okay? So I only have 4 questions. So okay. So here's a couple questions. I'll give you guys a minute for each question. No, they're not. They're not. We couldn't get the thing to work for some reason, so you don't have to worry about it. Does everyone have an idea of what they think it is? What do y'all think it is? C. Okay, good. So try to hammer that home to you guys that it's not that. So hypoblast does not make the chorion. The hypoblast makes the lining of the yolk sac. Okay? So the yolk sac, remember the pictures that we had of the yolk sac? That's made by hypoblast. Anything else on here is labeled besides that wasn't part of the answer. Okay. These are the kind of answers they usually give you that are trying to confuse you. But here's another question. Straightforward. Anyone have an idea of what this is? Okay, alright, good. Hey Alice, the one I told you about that you didn't care. I don't know why you just mentioned it. Here's another question. So he's likely to ask you this question, but he won't ask you the first part. I just threw it in there to make sure you guys understood this. have two inner cell masses, because early formation of blasts, trophoblast forms, then internal cell mass, inner cell mass starts to form, but it's split right before the inner cell mass happens, so you'll have one outer cell mass, but two inner cell masses. So you get two of these, so you can already throw out B and D. You can already throw out B and D also because I told you guys that if you have ICM number, you should associate that with amnion number, because the amnion comes from the ICM, okay? So it's so easy to get it. And then you'll have one chorion and one placenta, because a lot of those layers come from what's the extra embryonic stuff that was made from the trophoblast, okay? Which you only have one of, because it was formed already before the split happened. Cool? All right, one more question. You guys think? D. So epiblast made the extraembryonic mesodermal cells. Okay? The extraembryonic endodermal cells were made from the hypoblast. Okay? Endoderm has not been formed yet. And then extraembryonic mesoderm, I told you guys, comes from the hypoblast. And if you guys, just in case, because a couple of you guys weren't as sure about this one as the other ones, but I'll show you really quick. Okay, so when this is forming, right here, hypoblast, let's see that, these are forming. The hypoblast is forming the primary yolk sac. These cells coming down right here are coming from the hypoblast itself. Those are the extraembryonic endodermal cells, hypoblast. And then this right here, where you have the chorionic cavity forming around it, comes from the epiblast, which are extraembryonic mesodermal cells. So mesodermal cells that are extraembryonic come from epiblast. Extraembryonic endodermal cells come from hypoblast. Okay? Anyone have any questions?
Would you repeat that last thing you said, please?
Yes. Extraembryonic mesodermal cells come from the epiblast. Extraembryonic endodermal cells come from the hypoblast. No questions? Is this helpful?